Okay, so this is Introduction to Viruses, Chapter 5, Part 1. So viruses are able to infect every type of cell: bacteria, algae, fungi, protozoa, plants, and animals. There are questions about viruses that still remain. There's lots of research being done in ways that we can treat them. Are they organisms? Are they alive? What are their distinctive characteristics? What is the connection between viruses and cancer? All right, lots of questions out there. As far as debating viruses, to deciding whether they're dead, deciding whether they're alive, are they active, are they inactive, we reference them as active or inactive. So viruses are not alive. The vital role of viruses in evolution. So these guys infect cells and can influence that cell's genetic makeup. So I'm going to say that a second time. Viruses have the ability to infect cells, and then they can influence that cell's genetic makeup. Viruses have shaped evolution of cells, tissues, bacteria, plants, and animals. 10% of the human genome, 10% of your genome, consists of sequences from viruses. 10 to 20% of the bacterial DNA that's out there contains viral sequences. So these guys, when we talk about viruses, they are obligate intracellular parasites. That means they cannot multiply unless they invade a specific host cell and instruct its genetic and metabolic machinery to make and release new viruses. In other words, they hijack a host and use its tools in order to multiply. So properties of viruses. I'm not going to read through all of this, but we're going to hit on most of this in this chapter. So make sure this is kind of like a cheat sheet, quick reference for viruses. So make sure that you read through this stuff on this slide. Okay, size comparison. So here's E. coli. We saw that in the lab. And when you look, here's a rabies virus. Here's HIV. Here's the influenza virus. Here's adenovirus, so poliovirus, yellow fever. Okay, so when you look at the difference between that and an E. coli, we can't see these under our microscopes. You have to have a very special microscope in order to view them. And of course, here's a yeast cell here. Components of a virus: they bear no resemblance to cells and lack any type of protein synthesizing machinery that's found in cells. They cannot perform protein synthesis on their own. The viral structure is composed of regular repeating subunits that give rise to their crystalline appearance, and they contain only parts needed to invade and control a host cell. So they are minimalists, right? They have an external coating. They have a core containing nucleic acid strands of either DNA or RNA. And some have one or two enzymes. So the viral components: we have a capsid, which is the protein shell that surrounds the nucleic acid. This is composed of subunits called capsomeres. The nucleocapsid is the capsid together with that nucleic acid. So you should know these terms. They're important. They'll come up on the exam. Naked viruses consist of only a nucleocapsid. Some have an envelope. So not all viruses have an envelope. That's an external covering of the nucleocapsid. So it's an additional covering. And usually that covering is a modified piece of the host's membrane. So it takes a piece of the host's membrane with it when it leaves the cell, which ends up being the envelope. Viruses also have spikes. They're found on naked or enveloped viruses. They project out just like a spike would be on maybe a dog collar. They project from the nucleocapsid or the the nucleocapsid or the envelope, and allow viruses to dock with the host cell. So those spikes are pivotal in the ability to infect cells. A virion is a fully formed virus that is able to establish an infection in a host. Okay, so a general structure of a virus. We have, you can see here's a naked virus and here's an enveloped virus. So we have the spike, we have the capsid, we have that nucleic acid, okay, like DNA or RNA. Okay, and here we have an envelope. Same things, okay, same sort of shape, okay, although some viruses are shaped different. And then this enveloped piece of it, which is actually, like I said, part of the host. It takes part of the host with it. So the capsid is the most prominent feature of a virus. It's composed of subunits called capsomeres. You can see if this is a capsomere, this whole thing is the capsid, and within that is the nucleic acid. Okay, an enveloped virus, like I said, takes a bit of the cell membrane of the host upon release. It can bud from the cell membrane, from the nuclear envelope, or from the ER, the endoplasmic reticulum. At the core of the virus, you're going to find the nucleic acid. It's the genome, or the sum of the total genetic information carried by an organism. Viruses, like I said, contain RNA or DNA, not both. It has one or the other. That genetic material can be single-stranded or double-stranded. It can be linear or it can be circular. The number of viral genes is small compared to cells. So hepatitis B virus only has 4 genes. Herpes virus has hundreds of genes. And viruses only need to be the bare minimum of genetic information in order to carry out their job. They have one specific job, and that is essentially to infect and destroy. I'm not going to ask you about what types are single versus double-stranded, but these are just some pictures if you want to look over that. More on nucleic acids. So RNA viruses can be double-stranded, but usually they are single-stranded. Retroviruses carry their own enzymes to create DNA from RNA, and that's known as reverse transcriptase. Basically, that allows the virus to make DNA from RNA. So if you think, okay, well, in our cells, the DNA has to be copied by RNA and taken out of the nucleus out to the cell parts in order to be made into, to read the code to be able to make proteins. If you have a virus that is an RNA virus, well, how do you copy the RNA? Well, the enzyme reverse transcriptase basically makes a DNA copy from that RNA. So it's kind of the opposite of what you would think that happens in us. And I just want to point out again, this reverse transcriptase allows that to happen. That's an enzyme that allows that to happen, or helps that to happen. Modes of viral replication. Viruses are like parasites that seize control of the genetic machinery of the cells. They're a hijacker. So the fact that they name things that mess up your computer a virus, it's a perfect example. That virus is useless until they put it into the computer, and then it takes over and it kind of hijacks the system itself. How a virus replicates dictates the way a virus is transmitted, what it actually does to the host, the immune defense responses in the host, and how we, the medical community, treat the viral infection. Okay, so I would expect you to come back in the regular PowerPoint for this and watch the flu video. It's kind of a fun and interesting video that I think you would enjoy. So multiplication cycles in the animal virus. So there are phases of the animal viral replication cycle. Adsorption, that's a D, A-D, adsorption, penetration, uncoating, synthesis, assembly, and release. Okay, you have to know those phases for the test. You are going to have to come up with them cold. They are not going to be listed anywhere on the test. And we're going to talk more about this as we go through this PowerPoint. The length of replication cycle varies from 8 hours in poliovirus to 36 hours in herpes virus. So I'm going to pause here for just a moment. Okay, sorry about that. So viral adsorption to a host cell membrane. So we have the host cell membrane, and these spikes need some sort of receptor to attach to. Okay, so and here you can see we have receptors on the host cell membrane, and those capsid spikes will attach to those receptors. If the receptor is not there, that spike cannot attach, therefore the virus cannot infect that cell. So when we talk about adsorption, there needs to be a complementary exact fit between the virus and the host in order for the invasion to occur. So like I said, the spikes must match those receptors. The fit determines the host range, who will be infected. So hepatitis B can only infect humans. Rabies can infect all mammals. Cells that lack compatible viral receptors are resistant to adsorption and invasion by that virus. So tropisms, it determines which cells will be infected. So hepatitis infects liver cells, but hepatitis is not going to infect, the cells of your foot, right? So mumps virus targets salivary glands, but the mumps doesn't infect the liver cells, okay? So when we're talking about adsorption, we're talking about an exact fit between the spikes and the receptors. And then that fit will determine whether it's going to infect you as a human or just a dog or, plants. And tropisms is specific to the cells, like which cells of that organism will they infect. The next step in the process of this virus infecting a cell is the penetration and encoding of that virus. So the flexible cell membrane of the host is penetrated by the whole virus or its nucleic acid. And penetration happens through endocytosis. So the entire virus is engulfed by the cell and enclosed into a vacuole or vesicle. So when those spikes match, the virus will then be engulfed by the cell. The actual encoding of the virus is when the enzymes in the vacuole dissolve the envelope, if there's one there, and the capsid, and then that virus fuses with the wall of the vesicle, and the viral nucleic acid is then released into the cytoplasm. So, and then suddenly you have, the nucleic acid of this virus rolling around your cell. Penetration through direct fusion. This is a different type of penetration. So direct fusion of the viral envelope with the host cell membrane. There are pictures in your book that illustrate this, all of this. So you should be following along in your book or with some sort of pictures in order to understand what's happening here. This occurs in influenza and mumps when we're talking about direct fusion of the viral envelope with the host cell membrane. The envelope itself merges directly with the cell membrane, and the nucleic capsid is released into the cell interior. Okay, so here's two ways in which the animal viruses penetrate. I don't know off the top of my head what page this is on in your book, but let me look. If you want a close-up and personal picture, it is on page 133 and 134. So there you have, on this side, you have the specific attachment is when the cell actually attaches to the cell membrane. It is engulfed into a vesicle, and then pieces of that virus are broken down, allowing the DNA to float freely in the cytoplasm. Same kind of thing here, just a slightly different process. It attaches. The spikes actually pull that virus into the cell and fuse. It's fusing the membrane of the virus and the membrane of, the envelope of the virus and the membrane of the cell, and then just letting in that DNA. Okay, there's a video here you're going to want to go back and watch in this chapter 5 PowerPoint. So synthesis, okay? Synthesis means you're building something, you're making something. So replication and protein production, okay? In DNA viruses, they enter the host cell's nucleus and are replicated and assembled there. In an RNA virus, they're replicated and assembled in the cytoplasm. So if you remember, DNA is in the nucleus and RNA is in the cytoplasm, and depending on what we're talking about, that can help you remember that a little bit. So retroviruses turn their RNA genomes into DNA using that reverse transcription.